Everyone. It's a pleasure to be here to see your faces. It's actually my first time in the beautiful city of Montreal, which coincides to be my first time attending the BOSC conference. Um, okay, so I'm just going to go straight right into my talk. I'm sure most of you have heard of antimicrobial resistance. It's not news to you. We know the AMR is a global threat, and there's been a lot of health care associated infections, which is rising as we progress in time. And recent estimates also shows that about 1.3 million deaths are associated with AMR globally, and this also been predicted that by 2025, so 2050, that's about 25 years from now, there could be about 10 million deaths associated with AMR, which is quite shocking. I really like this infographic because it shows you the picture of the deaths associated with AMR and other diseases and even road traffic accidents, and most especially cancer, which has a lot of attention these days. And with the prediction, it shows that the scale at which we see cancer deaths currently is what would see AMR in the future. And if unchecked, AMR could actually shave about 3.4 trillion US dollars from the GDP annually and push about 24 million people into extreme poverty in the next decade, which shows we have to take this really seriously. And traditionally, in the public health side of things, scientists would use what we call this diffusion test to actually test for the presence or absence of antimicrobial resistance. And this is true in growing the bacteria in the lab. And usually, what they would do is they would make use of what we call an agar plate, and then they'll have filter paper with antibiotics, positioned around the agar plate. And what they will do is trick the bacteria across the agar and let it grow for 24 hours. And this usually is like a gold standard and is usually very successful. And what we usually find is a zone of inhibition around the area where you put the filtered paper, and that shows that the antibiotics can actually kill the bacteria. But in some other cases, you can have resistant bacteria, just like the A, which I have here, where the bacteria would grow across the filter paper, which shows that it's resistant. And in as much as this has been successful and it's a gold standard, there's been some challenges across using this method because some labs might not be able to adopt this properly because they're just starting out, and reproducibility can actually be an issue in many cases. So we all know next generation sequencing has been really pivotal in the way we see pathogen genomics. And one example is the SARS-CoV-2 pandemic, which happened quite a few years ago. And scientists were able to monitor variants as they introduced into the community in real time. And we've also seen other cases where genomics has been used to monitor AMR outbreaks such as cholera, and even wastewater sampling that many labs are involved in these days, where they can actually see antimicrobial resistance in the community by taking a pool of sample from effluent. And many tools have actually been developed to actually predict and detect antimicrobial resistance in genomic data, and such as AMR Finder Plus, RGI, CARD. Some of you must have used these tools before if you're in this area of work. However, there's a challenge with this work because these only amplify or detect AMR in bacteria that are well studied, like E. coli, Salmonella, and Mycobacterium tuberculosis. However, there are some bacteria that are not well studied, and which is what I'll be talking about today, which is nontuberculous mycobacteria, or NTM for short. So this is a species of mycobacteria. They are understudied, and they usually found in the environment. So water you drink, even the soil, your soil around your garden, and even termites also. So they're everywhere basically. And they're opportunistic pathogens. Unlike Mycobacterium tuberculosis, the human-adapted pathogens, these are opportunistic and they can cause disease in people who are immunocompromised or even healthy people also. And the caveat is they're also difficult to treat due to the intrinsic AMR nature. So usually when people are infected with NTMs, they can be on long course of antibiotics for 12 months or even longer than that. And it's usually cases of reinfection of NTM, even after you treat them. So why do we really care about NTM? One of the reasons is this statistics from Public Health Ontario shows that about 73% of infections from NTMs are associated or caused by the avian species, and then followed by abscesses and many others like fortuitum, and there are quite a number of these as well. And it's on the rise, just like I mentioned at the start of my talk. There's been about 2.5-fold increase of NTM infections in Ontario alone. And on the global scale, I think about 4.5% increase every year is seen across the world. So this is quite interesting. And the antimicrobial resistance in NTM can be innate or acquired. Innate resistance is the inability of antibiotics to be taken up by the bacteria due to the thick mycelic cell wall or through natural occurring genes in the bacteria that can expel or prevent the antibiotics to actually kill the bacteria. Additionally, you could also have acquired resistance, which is through development of mutations in the drug targets and also movement or transfer of additional genes through horizontal gene transfer from other bacteria that have these genes in the environment through plasmids and etc. So I'm going to be moving on to ResFinder NTM, which is an expansion of ResFinder, which is an open access tool that's used for detection of antimicrobial resistance genes from genomic data. And what ResFinder does is it uses your raw sequence reads or even your assembled genomes and then tries to predict antimicrobial resistance genes and phenotypes from a collection of databases that's been built from studies that did phenotypic tests on this bacteria, and then they were able to see that these bacteria were resistant and these genes were present and are the reason for the resistance of this bacteria. And this database includes mutations. These are mutations that are in the target sites of the bacteria that then would cause resistance, and also genes that are associated with resistance. And this tool was published in Microbial Genomics, and you can have a look at it if you're interested in trying it out. So what we did with ResFinder NTM was to, not just look at what it does, which is Mycobacterium tuberculosis, aside from other bacteria that are currently in the database, because we know there are other mycobacteria that are associated with antimicrobial resistance and diseases in humans. So we decided to include, genes and mutations from these NTMs. So we did this by looking through peer-reviewed publications, those that have done phenotypic assays on the two most prominent species of NTMs, as avium and abscessus. And we looked at mutations in genes that are associated with drug resistance. And what we did was to curate this into a new expanded database for the ResFinder NTM. And this is currently hosted on GitHub, and it's undergoing continuous development. So we plan to include other clinical relevant NTMs into this. So if you're interested in trying this out, you can look at the QR code here on the screen. So the workflow is simple. So basically it uses the current ResFinder version. And here you have the gene and mutation database that are currently on ResFinder, and then we expanded the database by including those for avium and abscessus that we found were validated genes and mutations. And then these can take into 5 formats, which is the raw reads or your assembled genomes, using 2 different modes, that's schema and blast, to search for resistance or genes through mutations or the presence or absence of genes in a database. And this would then produce an AMR report which can be integrated into like a clinical laboratory information system or even surveillance reports for public health. So we also included a utility script that can help you merge your 100 reports into a single summary file. For those who have lots of samples and you want to look at everything in just a simple output file rather than going through hundreds of files. And also Like I mentioned, the database uses FASTA files of the genes, and also CSV file that contains the mutations such as indels and point mutations, and specific position where these mutations are, and relevant genes or ribosomal RNA that are associated with these mutations, or this resistance rather. And we also have the PubMed idea of the papers that have done this work, so that those who identified these mutations, you can go back to read the paper and then understand or see what they've done if you're interested. And newly added genes and mutations are also verified upon addition to the database. And what we did for the verification purposes was we took raw and assembled genome of the two species we had in this database, and then we passed it into the tool to test for accuracy. And we used two different file modes, the FASTQ and then the assembled genomes for this analysis. And what we noticed was that we had 100% sensitivity through the two detection modes. However, we did see a little bit of drop in the sensitivity rather for avium using the faster blast. And I think this was associated due to the quality of the assemblies that we had. The use cases for this is for clinical labs, those that have integrated genomics into their routine diagnosis. They can actually use this to, check for antimicrobial resistance in the isolates that they've grown in the lab or they've sequenced. And also public health labs who are undergoing genomic surveillance, and then research institutions as well. So in the future, we are looking at, including other NTMs, clinical relevant NTMs into this software, because we know that these NTMs are usually associated with respiratory infections and even skin infections also. So it's important for us to be able to monitor and see what genes are involved in involved in resistance of antibiotics. So I would like to end my talk here, and special thanks to my supervisor and our collaborators and my lab members. And I'm grateful to the BOSC organizers as well for allowing me to give a talk at this conference. Thank you very much, everyone.
Very quick question. So what are your approach for assessing the influence or significance of gene mutation, regarding to drug resistance? So have you taken into the binding site?
Have I taken what?
Binding site between the drug and the target.
No, we haven't taken binding site into that. So what we looked at are mutations that are present in the genes that have been validated through phenotypic assays. So people who've grown this bacteria in the lab, and they've tested them with drugs. And if, like the picture I showed, where you have the zone of clearance or zone of resistance, where they did realize that the bacteria is actually resistant to such drugs at certain concentrations. So that's what we actually look for.
So you are using the assay data part of assay validation. Yeah. So I was just wondering if you can take into account, knowing knowledge, about the drug, how drugs binds targets, so whether that will help.
Yeah. So for the future, we would obviously consider that into the mechanism of resistance, so people can know how the drugs or the bacteria are resistant to such drugs.
Yeah.
Thank you.